Everyone can hear me. My voice is amplified. I guess you should be here if you're a little stuck or you have some questions about how to kind of do some of this stuff. So, feel free to do it however you want and reinvent the wheel and all that stuff. I'm just going to kind of go over best practices, how lame people do this stuff, and then like standards, so that if you do come up with some cool findings, other people can work off of what you've done, so that you're not just like in your own world doing whatever you feel like. But you can do that if you want to. All right. Okay, so the Broad Institute are the people who generated ONO's data, and I would say they're the best place, arguably, to also kind of get an idea on the best practices. So if you go to that link, you can spend a few hours reading through all that. But this is just a really short summary of kind of what's happening and what the best practices will tend to cover. And so over here you just see like a quick overview of what happens in the sequencing process. Obviously we're somewhere today, this weekend we're somewhere between these two sections. And these are ideally, if you're submitting a paper to like Nature or somebody, this is ideally what you want to go through. You want to go through all these steps, otherwise, somebody somewhere who thinks they know better than you are going to call you out on it. So you guys should all have the BAM files, and if you want, you can extract the FASTQ files and do some of these pre-processing steps if you want, but it's really compute intensive, and I don't have a lot of faith that you'll finish by the end of the weekend if you decide to do that. So if you do decide to skip some of these best practices steps for the sake of time, it's totally cool, right? And then you can go back over the next few months and redo it using all of the best practices. So essentially, we'll just cover a little bit of these two areas right here, and there are no guarantees that these will give you good results, and I don't know if you can see this. There's a sad face and a happy face at the end of the best practices, and if you get the sad face, you just go back and change something and go over those best practices again. If you get the happy face, congratulations, you're done. Okay, so I guess there's been a lot of questions about how were the VCFs generated, what commands were used, et cetera, et cetera. Well, open up the VCF, and all along the top of the VCF, you have the header of the file. And in the header, it logs which commands were used. So for example, I highlighted a command here right below this line in the header that says format, and it says GATK command line dot apply recalibration dot 2. So this particular command was applied to generate this. This is one of the commands that was applied to generate this VCF file. And this is actually one line. I've highlighted, this whole highlighted what looks like a paragraph is just one line. It's just a really long command. Obviously, some of the directories and file paths in there aren't going to be the same as what you're going to be using. But if you parse this, just for example, this line in the header, you'll know all the commands related to doing a recalibration that generated this VCF file. And there's a bunch of these commands, right? The generation of a VCF file correctly, following best practices, there's a bunch of things that are used. So immediately afterwards, you see another command that was used and another command. So the VCF file, the header of the VCF file, before you get to the actual data, will actually tell you everything you need to know about how to make that exact VCF file or another VCF file in that exact same way. So you don't have to go look all over the internet and if you like talking to other people here, that's fine, but the real answer is already in the file. Okay, so that covers, I think, a question that people were having of what are the steps that were used to generate these data files. The steps that were used to generate them are in the header of the files themselves. All right. So then another question related to the VCF file a lot of people are having is which samples are in the VCF files, what are the samples? Same thing, if you look at the last line of the header, so not the last line of the VCF files themselves, the last line of the header of the VCF file. If you look at the very last line, and you look at the final columns of the last line of the header, and the number of columns aren't static, so the number of columns will change. So it's, I can't tell you like, look at column 9, because column 9 might not be the last column in the final line of the header. So in this, whichever file this one was, if we look at the last line of the header, the last few columns, every column after the column labeled format contains the name of a unique sample. So every column after format is each one of your samples. So in this file, for example, we have a sample called NA12878. We have a sample called OF, 1120, and we have a sample called VB1120. Ono is OF, because those are his initials. NA12878 is an industry-based standard. It's a Mormon lady from Utah, and everybody likes to sequence her. So if you just type in NA12878 into a search engine, you're going to get data generated on multiple platforms, analyzed using various kinds of algorithms. It's just a nice thing to have, and you can use it to kind of compare whatever it is that you're doing to what, it's the same file that everybody else is using to do their comparisons. So that's just a nice thing to have. So for example, this VCF has those three, and VB is an internal sample. So VB was run at the same time on the same machine that Ono's data was run on. So maybe that'll give you some other things to better gauge whether what you're looking at is artifacts or false positives, things like that. So I'll also cover like how can you correctly pull out just the samples you want, compare the different samples, smash things together, et cetera, et cetera. But these last two slides should have kind of given you an idea of how to go to the header of the VCF for questions that you might have. I'll go over how to read the header better in a second.
Quick question?
Yeah.
So the Mormon lady, is that the ref in this case that's being compared against, or how is her data?
No, the reference genome is first made by a group called the Genome Reference Consortium. It's a consortium, so it's fancy. And then it goes from there to UCSC, Santa Cruz. They mess with it a little bit. And then it goes somewhere else. So for example, look at the header of the VCF file. The second to last line in the header, it says the reference. What reference was used? It doesn't say reference equals NA12878 because that's not the reference that was used. The reference that was used was the Homo sapien assembly number 19. So when the Genome Reference Consortium releases a reference, they name it like 36, 37, 38. But then after UC Santa Cruz gets through with it, they name it like 18, 19. So 37 is 19. Sorry, that's not my fault. You gotta go talk with those people. And then once a company like Illumina or a place like The Broad or a place like Google gets a hold of it, they might even shuffle the reference around a little bit more because they might think it makes more sense to have the mitochondrial chromosome up top instead of chromosome number 1, or they might number their chromosomes as 0, 1 instead of 1, and that throws everything off. So it's important as you do your analysis to look at also the second to last line of the header to see which reference was used. You know, the general lesson is look at the header. But yeah, so NA12878 is not the reference genome here. NA12878 is just something else that was analyzed as a standard, as a control. You know, like in science, people are like, you have your positive controls, you have your negative controls, da 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 da. The reference isn't part of a positive or a negative control. The reference is part of the experiment itself. NA12878 is a part of your like positive, negative controls.
I saw that in the Google image there are the third VCF, which contain the header that called tumor and normal.
Okay.
What are they?
Look in the header. I don't have a screenshot of that loaded right now. I can go over that with you, but if you look in the header, it'll tell, if you look through the command lines, so I'm pretty sure that file went through a thing called Oncutator, which I'll go over here. Once it goes through Oncutator, in the header, Oncutator will be like tumor equals, and it'll have the name of the sample and the pathway, the file path for that sample. And normal equals, and it'll say in the header what tumor means and what normal means. So maybe just like Control F, you know, do like a quick search on that. Next one. All right, so the file specifications. Go here, you're gonna get the file specifications that you need. HTS means high throughput sequencing, which is actually what we're doing here. We're doing HTS stuff. And these are 3 things that should be important to you. HTS specs, if you go there, you're gonna get all the file specifications for like SAM files, BAM files, also VCF files. HTS lib is a C library for high throughput sequencing data formats. That's what you're dealing with here. So you should probably be using HTS lib, but it's okay if you don't use a C. You know, I don't know. And then SAM tools is just like a nice little example of what you can do with HTS lib, and you know, tools written in C using HTS lib for manipulation of next generation sequencing of data, which is what you're using here. So check this spot out. Here's an example of what the specifications actually look like. The specifications are released as PDF files every few months. So you can tell this looks like a VCF file because this is the specifications for a VCF file. And it's going through one particular scenario. You know, it's kind of like a mathematics book. They'll kind of tell you what's up, and then they'll show you examples of that demonstrate that particular, you know, whatever, the thing that they're trying to show. So this is just an example that they're showing, oh, if something is inserted between 2 break-ins of a DNA strand, this is what it ends up looking like in the VCF file, okay? But just go here, look through the specs, look through HTS lib. That'll really help you parse through the BAM files, the VCF files, and those are really the only 2 kinds of files that you should be needing to parse through this weekend, and it'll help you understand what you're seeing in those files. All right, next up. So, you know, there's just like so much stuff you could do. I wanted to give you guys some quick things that would be helpful for you today and tomorrow. There are obviously a lot more things you can do. So for example, because the VCF files tend to have multiple samples in them, so selecting variants is very important. Selecting variants from one specific sample is important. One sec. So you see it says select a subset of variants from a larger call set. So say you only want to select the samples from, variants from Ono's file, but you don't want the stuff from NA12878 or VB or whatever else is in there. Or say you just want the tumor data, you don't want the normal data. Follow these steps. It'll pick out just the variants that are for the tumor data or just the variants that are just for NA12878, and it'll give you only that data in a file. It's pretty easy. This isn't actually all you need to type in. You know, like you obviously need to like specify, you know, those things that you need to do with Java, like specify like memory heap or whatever. Like, oh, I use 8 gigs of memory to do this step. If you try to do this, you're going to error out, but essentially this is what you do. All the basic Java parameters that you need to like throw in there, you know, you can figure that out in like Stack Overflow or something. So let's look at these 2 examples real quick. You know, here this person is like, yeah, we're going to select some variants. This is the reference file, not an A12878. The reference file is either going to be something from the GRCH or UCSC. It'll say something like 19 on it. You know, Homo sapien 19 or UCSCHD 19. You know, V is the input VCF, which is the VCF you probably have in your cloud image, and the output VCF where you want this new VCF. SN is the samples that you're going to want, right? If you want the tumor, or if you want NA12878, something like that. Here, you know, getting a little bit more fancy. If you want to use a regular expression to pick out your selected variants and your samples, it's fine. It supports that, so you can, you know, come up with a nice looking regular expression, feed it that. You'll get the things that you want, and you won't get the things that you don't want. So if you do that, that should, I think, help a lot of you in terms of what you're dealing with, you know, which sample is what, which variants belong to what sample. You can do this to separate everything out and kind of sift through the data that way. Variant annotator, I think, is going to be helpful for you because it's going to allow you to add information from other places and sort of contextualize the data that you're looking at. Again, here's some quick examples. This isn't exactly what you need to type into the command line. If you type this into the command line, it won't work because you need to, you know, type in all the parameters for Java for it to work. But essentially, so you know, here you're using variant annotator. Now you're going to need the BAM files if you're going to use something like variant annotator, because you see one of the requirements is you need the BAM file, and then. Over here, this person, in this example, the variants are being annotated with a database called dbSNP, which the US government hosts. It's not a cancer-specific database, but just for this example, it's a good database to use. So you see at the end they're specifying that it's dbSNP, and this is how you do it. Let's see, and here, in this next example, allele frequency. So alleles are like, you know, I don't know, it's a weird word, it's hard to say, it's hard to spell. Those are like, you know, is it an A, is it a T, is it a AT, whatever. Those are alleles. So where are the frequencies of those things happening? Again, you can do this with variant annotator. It's kind of, you don't have to do this stuff with variant annotator, but like I'm saying, like, I'm just showing you like industry standards and best practices. Here they're doing a quick example of how to, you know, sort out your data and pick the things based on allele frequency. Like, oh my God, I only want the things that say like G more than 90% of the time or something like that. You know, make up your command, throw it in there, it'll spit out the file that you want. Variant filtration. Okay, it's kind of like variant annotator, but now you're actually filtering because variant annotator isn't going to make your files shrink. Variant annotator is only going to cause your files to increase in size. Every file that you have, once you go through something like variant annotator, because it's going out into the world and picking other things to contextualize the information in your file with the databases that exist in the outside world. So that's going to cause your files to swell up. And you know, if you're dealing with a signal to noise or a needle in a haystack problem, it's going to seem like you just made life worse for yourself because instead of having less data at the end of the day, now you have even more data. What the hell? So variant filtration is going to be helpful because, and I would run it in this order. I wouldn't run it in some mix. I wouldn't filter first and then annotate and then select the variants. I mean, you could, but I don't know what you're going to get if you do that. So variant filtration, is going to help you shrink your files by filtering stuff that you don't actually want. I know I said like variant annotator is going to not show you stuff you actually don't want. It's going to show you everything. It'll just put a little word next to the things that you don't want. It won't actually get rid of them. So you can use variant filtration to do that. Variant filtration is pretty interesting actually because it uses this thing called I think it's JEXL, like the Java expression language. So you can kind of read through any given line in a VCF file and try to construct an expression that makes sense to you, like an expression that's going to get rid of the stuff and keep the stuff that you want to get rid of and keep the stuff that you want to keep. So this example, this person is, I don't know, I guess they're going to keep stuff where the AB of the thing, whatever AB means, equals less than 0.2, or the MQ0 of the thing is higher than 50. So just a quick thing to remember with these expressions is once you write the expression, the immediate name that you apply, so you'll write the expression, then space, and then filter name, you're going to give it a name. That name is that expression. The file itself won't write into the file the expression that you wrote. That your expression is going to get written into this name. So for example, in this person's case, they wrote this expression and it's just called November 9 filters. So in the file, if you actually search for this expression, you won't find it anywhere in the file, but if you search for November 9 filters, now it will. So if you're getting fancy and you're like, well, I'm going to do like 4 different expressions, okay, but just remember that the order that the expressions will be named will be the order in which you entered the expressions. If you try to play with this, you're probably going to run into problems, but now you know what the solution is. Okay, so far everything we've gone over isn't specific to cancer stuff. So let's go over some stuff that's actually, made for cancer. I don't have to read this for you. You can read this. It's reliable and accurate identification of somatic mutations and cancer genomes. So people use this stuff a lot, and it gets good. The journals like it if you use this stuff. So this kind of goes together like mutec, mutsig. Look that up if you feel like using it. Again, I don't know how much computational power it's going to take. Not in terms of like, do we lack the power, but in terms of do we lack the time? Because I don't know if you'll be able to finish it in time for Sunday night. But at least now you know like this is the right algorithm to use.
Can I ask a question?
Yes.
So I was talking to one of the teams about somatic variant calling in split VCFs, and we looked at mutect, but it seems like polymut might be a little bit easier for them to leverage.
That's fine.
Do you have like particular thoughts about like the different tools? Because like I kind of thought it was somewhat all the same. It just seemed easier.
The way I like to do it is I'll type the name in of an algorithm or a tool on Google, and then I'll click on similar, you get your sort, and then I'll get a list of all the other ones that are, because what happens is like, kids are getting their like their PhD, they're doing their dissertations, their postdocs and stuff, and everybody wants to write their own little algorithm and tool, and so there's quite a bit of proliferation on that. Yeah, I just use this because I think this probably has the most citations in that field, but doesn't mean it's the best.
What you're saying, it's not really that.
You can pick, yeah.
Cool, thank you.
Yeah, exactly. So, like I said, is this the best algorithm? I don't know, but nature thinks it is. Not nature as in the universe, nature the magazine thinks it is. And like the guy who made it, some of you might be really a lot better at mathematics than I am, so the guy who made it put in a lot of work doing mathematical proofs of why his thing is the best. And I don't know if the other algorithm the gentleman just mentioned, if that guy put in the, all the work of proving why mathematically his algorithm is superior or whatever, but this guy did. So, maybe that's why the magazines really like him. a lot more than everyone else, even if the algorithm itself doesn't perform as well. So yeah, if you're interested in doing something like that one team was going to do, then yeah, go ahead, check that out, or one of the other ones. All right, so HTSlib, really super important, maybe not, you can find other ways to do it. HTSlib is just the standard, it's the fastest, the guy who makes it is like, you know, a god amongst men, etc. So yeah, it'll let you look through your SAM files, BAM files, this new format they're trying to peddle called CRAM, and your VCF files. One thing you can do really quickly is, you know, just go look at your VCF file a little bit. It's nice to visually see things sometimes, not spend all your time in like columns and rows, and I mean, this is still going to be inside of a terminal. Yes?
You're going to post this on the Slack channel?
I think it's already on there.
It is.
Good, thanks.
And here's a couple of, so you know, if you go on there, there's just going to be not just an HTSlib, but also in the SAM tools implementation of the library, there's just going to be a lot of options and things, and I don't know if you're going to have time to go through all those, so I just quickly you know, put the command that I think would be helpful for you here. It's tview, you know, it's pretty simple. This will show you how to use it. So just, you know, if you go onto the documentation, just Control F tview and then read how to do it and then do it and be happy. Here's some other examples of stuff that should be fun for you to do. Because like BAM files are difficult to work with because they're binary, you know, so it's much better if you if they're not binary anymore, then you can actually you know, make some use of it. So I just took a little example of how to make them not binary. When they're not binary, they're called SAM files. So there you go. This is the way to do it. That might probably make life a little bit easier for you, but your data is gonna really blow up as soon as you do this. The reason it's binary is for cost and efficiency purposes. So once you make it not binary, it's gonna get really huge and difficult to move around and expensive and all those other things. So HTSlib, go check it out. All right, and then some other tools that could be helpful to check out, you know. I think I mentioned all of these already, maybe not, you know. But these are pretty standard. It would be ridiculous if you're like doing this stuff and you're unaware of the cancer genome atlas. So, you know, at least make yourself a little, at least in your browser history, have it so you've gone there and looked at it, you know. So Cosmic is pretty good, SNPF is pretty good, Longitator is pretty good. It's a little bit difficult to, all these things also require a lot of additional files, and this is just an example, like the Oncitator, you're gonna need its corpus download, which is 17 gigs. You know, and all these things are highly experimental because we're in experimental science. So there's going to be lots of bugs, lots of things aren't going to work, and you know, you're just going to have to figure it out because that's the space you're in. So that's all the questions that I could think of that you guys would have, but you can ask me other questions that you've gotten stuck at. But like I said, the headers and some of these locations I showed should be your main resource for figuring out how to do things correctly. That's all.
If there are any questions, I will go around with the mic.
Hi, I got a question about the reference you're using for VCF. It's HG19.
Yeah, it is HG19, but what happens is the Broad likes to take HG19, change it up a little bit. So if you try to use the HG19 that UC Santa Cruz produces, things are going to break and you're going to get errors. So did we provide, we provided the reference on
Yeah, I saw
the
that.
image.
And do you think you can provide us like updated version like HG38, which is a standard version right now?
I'll tell you why you shouldn't use that, because the alignment uses a reference, right? So to generate the BAM files, you have to use a reference. So the BAM files, the reference that's provided for you is the same reference that was used to generate the BAM files. If you use a different reference file, there's no guarantee that the BAM files that you have access to will be compatible with it.
Yeah, you lost some information around the telomere region, but I'm still thinking about there are a lot of biomedical tools very good compatible with HG38. I think is there any easy way we can lift over version from?
They do make some leftover tools. If you go to the NIH, or no, if you go to NCBI, right, the whatever that means, it's a sub-branch of the NIH. They make a leftover tool. However, I'm going to tell you, all these other steps and algorithms and things don't support usage of a leftover tool. So if you use a leftover tool and you're getting bugs and errors and things aren't working, no one's going to be there to have your back.
Yeah, I think that'll be hackathon. So, how about like if you can just from BAM to extract all the reads, raw reads out to the FASTQ file, then just read line with HG.
Yeah, you can do that. I don't think you can do that in two. I mean, you can do that, and then I don't know how long it's going to take the computers to finish that realignment.
Yeah, that's how I'm trying to access like some function like HG.
Yeah.
I'm stuck somewhere by the function.
I can come look at, or we can kind of see what you're thinking, but I'm probably going to try to talk you into not doing that stuff, because it's going to take weeks of just running it in the cloud.
That's right.
Right? You're not going to finish it today, you're not going to finish it tomorrow, you're not going to finish it next week.
Yeah, thank you.
That's what I'm saying. I'll come.
More questions? There must be some.
His questions don't count.
Yeah, it's just a general question, but do you have any, not totally related also, do you have any tips on how to turn this VCF into an actual potential-like data?
More what? What did you say?
Tensor, like a matrix form.
Yeah. There's a library for that. I'll show you the library.
Do you know if there was single or parent sequencing performed on the sequence?
If there was single or parent
sequencing.
I think it was parent.
Okay, cool.
I think so. If you look in the BAM file, if you turn the BAM file into a SAM file, and then you kind of take a good look at the BAM file, you should be able to tell 100% for sure which way it was.
More questions?
So we have two VCF files. Could you, because I noticed the sample name for each of the two, that's down. So we have OF, I think. Could you explain the headers of those two VCF files to us? What I know there's like data information there and like tissue sample, tissue type.
Yeah, so OF 1120 is Ono's blood. And then there's another OF sample name, which I believe ends in _A, so like OF, some numbers like 01 something something -A. That's his tumor. Then there's another sample called NF2XY_S, that's his sibling, who's a male, XY. All the other samples are sort of standards and controls for you to kind of play with. If you guys have like really specific questions, you can, track me down or something.
All right. That's it.
I think there's one up here.
Thank you.
Yeah, welcome. You guys can bug in if you have more questions.